Aloha, everyone. Thank you so much for being here tonight. Let's see how this works. Is it the arrow button that advances the slides? Spacebar. Okay. Let's see. Yep, I'm Michelle. Aloha. How did I begin this? Yes. While my title suggests that I've been working with textiles, I've done that for a while, but I haven't always done that. So I want to give a little bit of background to give some context for how I arrived at this gig. When I grew up in Wisconsin, I was asked, what do I want to be when I grow up? And I want to be an oceanographer and a marine biologist and an abstract artist. And while I reflected upon this recently, it occurred to me, that is crazy. I mean, I was always doing realistic renderings of things in my world. So I was doing very representational artwork. Where did this abstract thing come from? I don't know. And then a marine biologist, I was in Wisconsin. I was landlocked. I had never been to the coast. So I don't know where that idea came from either. But reflecting upon that now, it occurred to me that I've brought those things around in my life. And I'm really glad that this Artist at Sea program has enabled me to see the full circle that has been closed through this program. So in college, I decided I would go to school for science. I thought that was practical. My curiosity was always piqued by the natural world. And I studied entomology and wildlife ecology. So learning about conservation biology, I learned about a lot of Hawai'i species, but I didn't know that I would ever end up here. But as I was learning things, I would often draw pictures of the things that, the anatomical structures I needed to learn about. And then one of my professors saw my drawings and said, hey, would you like to work as my science illustrator in my lab? That blew my mind. That became the job that put me through college, and I continued to work as a science illustrator for about 15 years. Now, let's see. I moved to Hawaii in 2005. I became a parent in 2008. At that point, I had started to think about do I want to remain an illustrator all my life, or do I want to branch into more of the fine art aspect of creativity? And at that time, I was toying with the idea of maybe a master's in fine arts, but then to my surprise, I was invited to be an artist in residence here at UH Manoa, and in the Department of Fiber Art. Now, the reason for this was in part because I had started some community art projects using knit and crochet as the medium that people would work in. So that was interesting. Let's see, I have some pictures. I always forget to start using the slides. So here are some of the illustrations that I did over the years. But then what culminated at UH was this project. It was an installation at the Commons Gallery, which utilized fiber and found objects to recreate the Hadal Zone. If any of you were here for Jeff Drazen's talk a few weeks ago, he's the person who had a lot of the specimens that I worked on. So it was fantastic to know that within the university I could go and reference another department, again, doing a collaboration with scientists that kept me learning on the job, but also communicated science in a way that was accessible to people, in a way that it might not have been if I had been using another medium, perhaps glass or metal sculpture or something like this. So as I'm knitting things, it seems like people are more willing to engage with these subjects. That's a life-size amphipod. It's crazy big, by the way. And then the professor that invited me to UH sent me an email. It was saying that 2 days from that moment, there was a deadline that she thought I should go for. And the deadline was for this Artist at Sea program. I had not heard of this, although I had heard of the ship. The voyage that Jeff Drazen had been on to collect those amphipods and those snailfish, he was on the Falkor. So when I first learned about the Falkor, I thought, whoa, if only I had stayed in science, if only I had, continued to be a field researcher, maybe in marine biology, I might have been on that ship. But as it turned out, here was another way for me to get on the ship to collaborate with scientists. And so I applied. 2 days later, I was accepted. 9 days later, I had to be ready to leave, to be gone for a month. That was crazy. But luckily, my family helped me tremendously, and away I went. But since there are no yarn stores at sea, I had to gather my materials ahead of time. And I knew a little bit about what kind of science was going to be happening on board, and some of it was about proteins, about proteomics, biogeochemistry. A bunch of these terms were flying around, and I barely had time to figure out what I was going to be working with, but I knew it was about data, and I knew it was about protein and water. So the yarns that I got here were from the Big Island. It turns out there's a ranch that has Romney sheep on the Big Island, so I gathered these yarns in a nice gradient of natural colors. And I packed up my suitcase full of yarn and headed to the ship. The first day on board, I had to get my sea legs, and I wanted to create a relationship with the ship. And though I didn't apply as a painter, I did bring some paints, I have to admit. This is a little demonstration or a little example of the setup that I had. Each day, I wanted to do something each day that I was on board. And in this case, I loaded the paintbrush with a certain type of paint. It had micaceous iron oxide in it, so that related to the structure of the ship. And I let the paintbrush work like a pendulum and move across the single page. Whatever the ship's heave and pitch and yaw was doing, it would end up on the paper like that. So for 1 minute, I did one of those a day. And this was a sequence of a few of the days. I was so pleased that the voyage that I was assigned to left from Honolulu, that it was coming from here. This is my home. This was a fantastic way for me to learn more about my home by leaving my home, but to put it in context of the Pacific. That was very powerful for me. On the route, you can see Honolulu is on the top, going toward Tahiti. Something I did understand about the voyage was that the main action done by the scientists would be to collect water samples. And in this slide, you can see one of the examples of the equipment that they would use to collect those water samples. The gray tubes that you see are open in this case. So the equipment, the rosette, would be lowered down into the water column. And let's see, here they're closed. So I think this is showing one of the students collecting some of the water samples after it's been utilized in the water column. So at night, or wherever we would be, wherever our predetermined stop would be, the ship would stop, and then the equipment would be lowered down on that wire, on that cable. Remember the word wire. And then these are the types of data that the scientists would be looking at in the control room. The center top graph is what was of most interest to me. And let's see, the way to orient yourself about this, look at the y-axis, and that is an indication of the 0 level is the air, water, surface, and then as you go deeper, it's going to be lowered down to whatever depth the scientists want it to go to. Once it's down there, or as it's going down there, the green and the blue and the purple lines are showing up. So those are data coming in that are provided by the sensors. In this case, the sensors are showing us the green is light level, the blue is oxygen level, and the purple is the temperature of the water column at that place. So again, the word water column was a phrase that I was hearing a lot, and I was latching onto that, wondering how I would use that in my work. And then each of the horizontal bars that you see, those yellow lines, indicate where the scientists would use their mouse to click a button, and then the gray container that you saw, the lid on one of those tubes, the lid on the top and bottom would close, and then that would provide a discrete water sample from a specified known depth in the water at this specified location. So this was the action that the scientists were doing on board. They weren't collecting organisms, they weren't using remotely operated vehicles. You know, that was what I thought I was going to be getting into here, but not so much, but it's okay. Luckily, in my science illustration training, I learned about information graphics as well as rendering organisms. So the idea of working with these data sets was appealing to me. I was glad to be able to have this challenge. Now I got to work. I started using my knitting machine. The gray bars that you see here have a series of hooks in between them. And if any of you ever did latch hook rugs, that's the action of the wire that's in there that helps create each of the stitches. So my knitted section is hanging down below. Up above you can see the wool yarn. The blues are threads of silk that have been dyed with indigo. So I was happy to have that yarn in my stash already, and I brought it on board realizing this is the project that it's been waiting to be used in. The square would be a swatch looking like the depth, and then on top of the swatch, I crocheted the data in colors similar to what you saw on the screen. I was learning from the scientists that these data sets would likely never be published, but it was the very important information that they were getting in the field that would help them make their decisions about where they would take their water samples and then be able to place the information that they would later learn from their analyses in context of the Pacific Ocean. I ended up doing a series of these for every station that we stopped at along the voyage. These are just the first 4. But all the while, I was thinking about how I could take these profiles. The lines of the data set are also called profiles, like the oxygen profile. Think of it like the outline of your face. But then I started thinking about how I could move that around in space and then have that be a limit for the way I would create another kind of sculpture. So this is a first generation idea. Where would it go? But at the same time on the ship, I also had a little side project. Can you all keep a secret? Well, I may look like a regular soccer mom, but I'm also a graffiti artist. And sometimes I have been known to be involved with yarn bombing. And there's a form of graffiti where people will knit textiles and then put them up out in public, usually without permission. And this idea was in my head. And when I learned about naval history, in World War I, the British fleet was often painted in this picture down below. This crazy, bold graphic stripe pattern is known as dazzle camouflage. And hard to believe, I know, but in the early 20th century, that was effective camouflage out at sea. So I ran with that idea. I thought, I want to make some dazzle camouflage textiles and put them up on the ship while we're out at sea, right? So it's this mobile installation, mobile graffiti intervention almost. So up above, yes, you can see that on the Falkor, there are lots of cameras watching out for things to always be going right. So they probably saw that, they probably figured out who was doing this, but it was still worth it. Here are a few examples of the things that I did on board. And these are also, as Carly said, things that can be seen at the Bishop Museum exhibit. So come on over and check them out. So once the voyage was over, I flew back home, and it occurred to me that the route that I had just sailed was tracing the route of the Hokulea voyages, right? Between Tahiti and Honolulu. It was also tracing the path of Polynesian voyagers from centuries ago. And it occurred to me that this is another story that I somehow want to layer into my work. I didn't feel like my work was done once the voyage was done. This experience felt so generative. It felt like I would make multiple versions of work, multiple series of explorations through this experience, and it's come to be so. If any of you heard about the Honolulu Biennial, and if you went to the hub, you might have seen some of the work that I did there. I was very pleased to know one of the curators from that, and she knew about my voyage. She asked for a studio visit afterwards, and once I met with her and told her what my experience was like, and especially the link to the region we're in the Pacific, in Polynesia, that was definitely of interest to her. So she commissioned me, or they invited me to do work that would be displayed in the biennial, and it ended up being my largest solo works to date. So I'm very pleased that I had the opportunity to do that. Thank you, Falkor. The work that I was proposing to do was not necessarily something I could keep doing with fiber, traditional fiber as we know it. Instead, I started thinking about the materials that I was seeing on the ship. I thought maybe I need to work in a material that has more structure. So it occurred to me that equipment was going down on a wire. And I thought, I've knitted with wire before, so why don't I keep knitting with wire? Made some samples, the prototypes were accepted. So these are two of the finished pieces that referenced the CTD data. Again, the original squares that I made, I would take three of them, or I took one from a different location and then spun it around the y-axis. And the levels that we saw of the data became the limits for how I sculpted these pieces. They have traditional knitting techniques in them, but they are not necessarily done with needles. We can talk a little bit about that later with the show and tell. But I'm so excited that this voyage continues on for me. I feel like I still have inspiration that has yet to be tapped, and it has opened some serious avenues of interest for me. So if you can read the top of that shirt, my sister gave this to me. She knows me well. The shirt is a spoof on a Dr. Seuss poem, and the first line is, "I will knit on a boat." And indeed I did. So there's the Falkor. All right, I will now turn it over to my fellow science communicator, Kirsten Carlson. Are you ready?
So my name is pronounced Kirsten, not Kristen. On the ship, I saw my name spelled lots of different ways, including Kirsten, including Kristen, Christine. It was awesome. It's a common issue, so I just clear that up right away. When I was invited to go on the Falkor, I got the opportunity to go on a plankton expedition. So I am a science communicator by way of being a scientist. I have an undergraduate from the University of Missouri in biology with an ecology emphasis. And then I transferred out to California because I wanted to be a marine biologist. And out there, I attended Moss Landing Marine Laboratories, dropped out because I went to a place called Antarctica and did marine science and realized I wanted to convey the beauty and wonder of nature to people more so than research. And because I'd always been artistic. So I work with lots of different audiences to produce things for lots of diverse groups of people. For example, this is a graphic, an infographic I did for a scientist for presentation. I did new species illustrations for I'm still doing species illustrations for publication for scientists. Government agencies to products like this poster I did for the Marine Sanctuary in California, their goal was to educate the public about the two sides of nature. There's good stories and bad stories, and so they wanted to highlight the threatened and thriving species of Monterey Bay National Marine Sanctuary. Illustration and design. So I came up with the illustration concept and the design concept. They're still being used today. They're a free giveaway, and it was for the general public. This is a very recent project that I've been working on where I'm evolving the idea of adult coloring books into the idea that you can use art and science together to educate yourself about nature. And the series is called Artsopedia, and I have a fond place in my heart for plankton. I have since I was an undergrad in Missouri when I found out that larval forms were named completely different things because the scientists at the time in the 17 and 1800s had no idea that these creatures grew up into other things. They thought they were separate species. So this is the inside. All of it's colorable. On the inside, you learn a little bit about the 4 major types of plankton. There are tons of ways to categorize plankton, but the coolest part is I went analog on this. So when you're coloring it, you rotate it to find out more about plankton. I also work on children's books. So a huge audience of mine is working with kids and those that are young at heart, because I'm honestly still an 8-year-old at heart, about concepts in nature. I work on a lot of nonfiction books. I have done 4, 3 as an illustrator, 1 as a illustrator writer. And I'm currently in production on several written and illustrated books by me. The core theme of all of these, other than there's a plankton theme, I don't know if you caught them all, but it's my goal to connect people with nature. And at the core of that is actually understanding nature through science. But where I took off from science, because I went into science communication, is I wanted to add some additional creativity and imagination in the form of art. Science takes a lot of creativity and imagination too, I'm sure you all know. But the reason I wanted to do that is because there's threefold reasons. One is using art and science together increases the accessibility of scientific concepts and nature to lots of different audiences. You just saw some examples of how I've done that with my art. Visualization is a way to grab people quickly. In this information overload age, it's super important to convey stuff quickly. Images convey information thousands of times faster than a block paragraph of the same information. Hopefully by the end of this, you'll have seen that from both Michelle and I. And the big picture is the idea that everything is interrelated. The father of ecology, somebody that I just recently found out about, named Alexander von Humboldt, actually he is quoted as saying, everything is interrelated. He was kind of vanquished from our records because he was German and he was alive during the time of Napoleon. So when World War I and World War II rolled around, he was kind of ex-nayed from our history books. He's the result thing. He influenced Darwin. He influenced Thoreau. When I was reading a recent biography about him, I found out a lot. The book is up here and you can look at it later. So how was I going to take this concept of understanding nature and apply it to the stories I wanted to tell about this particular cruise with these scientists, the Sea to Space particle investigation, which happened earlier this year. On this cruise, there were NASA scientists and scientists from all over the globe whose focus was to take the big picture of the globe and some of the satellite imagery that NASA actually takes of our own planet. They don't always look outward towards space. They actually look at our oceans. And combine it with oceanographic data that is the micro scale. We were transiting across the Pacific Ocean in an effort to compare and contrast the satellite imagery and what it was measuring with actual measurements from the plankton and other tiny particles in the ocean. Interestingly enough, the hashtag is Sea to Space. So what was unique about this project that Michelle and I, it factored into what we needed to do, is that you need to finish your art project within the confines of the cruise. That's unique for this, to me, for this program, because a lot of the products that you saw take me weeks, months to complete. I'm sitting in my studio drinking a latte. I'm not on a pitching and rolling ship. I had a few days short of 4 weeks to produce these concepts or produce these products that reflected the story of the Sea to Space particle investigation. So because I'm an ex-scientist, as I like to say, I was fascinated by the fact that I got to go on this trip as an artist. And it reminded me of the days 200 years ago when the scientists brought with them naturalists and artists to actually record the information that they discovered. So I am fans of many of these people who I will show, and some of the books are up here. But the reason that I wanted to focus on this is because it gave me an opportunity to focus on the contrast between 18th century science communication, 21st and 21st century, 18th and 21st century technology, and 18th and 21st century scientific theory. So this is an image that shows some of my idols of yesteryear. So at the top is Ernst Haeckel. He has done magnificent drawings of plankton and other things, actually. If you haven't seen his stuff yet, I suggest you look at one of the books up afterwards. I can't even draw close to the man. William Beebe, he's one of the first people to go to the depths of the ocean and describe what he saw for a female artist to paint. And then Alexander von Humboldt. Again, I recently discovered him and was fascinated that he was the guy who came up with a lot of ecological principles that I learned when I was going to University of Missouri as an undergrad. So I'm a traditional artist. That means I use watercolors and I use pen and ink. I use something called Scratchboard, and I sometimes use my iPad and iPencil. So I do stretch to the 21st century with my technology. I use digital a lot when I'm doing design work. So my background as an illustrator and a designer incorporates lots of these elements. My backbone to this trip was going to be my sketchbook, which is also up here on the table, and you're welcome to page through it. I brought some samples that you guys can play with. If you've never used a water brush pen, they're pretty fun. So with this idea of understanding nature and wanting to make stuff accessible, I wanted to figure out how I could tell the story of what the scientists are doing while they were on the ship. They were using lots of new technology. Well, fortunately, on the ship, we had a fabulous communication person, Monica, and she and I collaborated together to produce an animation that explained what the scientists were doing. So I want to take a valuable 3 minutes of my presentation time to show you this, because I think it did a stellar job of showcasing how art can make science accessible through story. Oh, sorry, I forgot one thing. I was changing slides around at the last minute, always a dangerous thing. So this is my sketchbook. Like I told you, this is my backbone. And the thing about the sketchbook is I am working rapidly. Sometimes it ain't pretty. And if you can see what's written in the lower right-hand side, it says, "Big waves are sliding my chair across the room. Watercolors with waves get messy." So I was doing this during a talk by our lead scientist, Ivana. And the reason I wanted to show you this is because I simplified what she was talking about into this visual. The big picture, little picture of what the scientists were doing was the satellite imagery on the big scale, the phytoplankton on the small scale, and what they were comparing was quality versus quantity. Satellite imagery can take big snapshots, but sometimes the resolution isn't very good. And when you're on a ship, sampling like we were, with the equipment I could tell you when and where I saw what plankton in our transect across the Pacific. It's one of the coolest things for me to ever experience because I had never in my life been able to see plankton with my own eyes. The other really important thing I want to stress here is you see how phytoplankton's at the top, satellite imagery is at the bottom, and the carbon cycle is a really important aspect of this. It's off to the left, and the satellite imagery and the oceanographic data that they were taking in is trying to look at the global ecosystem of the carbon cycling system, the global cycle of carbon. I still can't say the right, you got me though, carbon cycling. Because it's really all about the phytoplankton and how they take up oxygen, how they produce oxygen. Okay. So this is the collaboration I did.
Field sketching on a boat like this is full of challenges to deal with the pitching and rolling of the ship. That is a really small price to pay though, because the equipment that the scientists are using is amazing. So I'm drawing this equipment in my sketchbook, and I'm hoping to take you on a tour of it with these drawings.
So in order to understand more about our planet's carbon cycle, we need to understand more about the production of oxygen through photosynthesis and also the consumption of oxygen through respiration of microorganisms. So we're doing this using a unique instrument called a photosynthetron. The surface water samples are exposed to different light conditions that simulate different depths and time of day. Through these filters, we can measure oxygen production during the daylight and oxygen consumption at nighttime.
Understanding whether particles near the surface of the ocean are big or small has implications in the carbon cycle. An instrument that measures particle size as we pump seawater through it. Inside the instrument, a laser beam passes through the seawater sample that we're pumping, and all the particles suspended in the beam scatter light in different directions. We're able to then measure that scattered light to tell us about the particle sizes in our sample.
There are many things we need to measure, not just particle size and distribution. The physical characteristics of the water column are also very important. One tool we use is the wire walker, which drifts for days carrying a whole array of instruments. A buoy sends a GPS signal, and down below the surface, our walker goes up and down a 125-meter long cable, measuring temperature, salinity, oxygen, and sunlight. We also measure fluorescence and use several optical instruments to understand how many particles are suspended in the water. Finally, below the walker, a sediment trap catches falling particles that we can analyze later.
One of the most exciting technologies we're using on board is virtual reality. A holographic camera dipped 1/100th of a meter below the surface allows us to create these experiences. As particles pass through an opening in the camera, a laser wave interacts with them and stores the information as holograms. You can later use the wavefront information to reconstruct the different particles, shapes, and positions in the water. That information can be displayed in virtual reality afterwards, which allows the scientists to interact and study these microscopic organisms in a whole new way.
When you develop a new technology, there's always risks. What we're doing here, although we are developing new technology, we're doing the same measurements in traditional methods. So even if the new technology fails, we're always going to be able to rely on the traditional methods. And we can use these traditional methods to kind of like calibrate, see if these new technologies are actually performing well. So, you know, in worst case scenario, we have something to fall back to, but so far so good. Everything's been great, so we'll see.
Thanks. So I have to tell you that that's one of the stellar things about the Schmidt Ocean Institute that I was so excited about, is they put a lot of effort into putting science communication out there on the fly. So this is a little bit different than the scientists and artists of the 18th century. This was done while we were underway, and on both Michelle's cruise and my cruise, you guys still have access to that even though the cruises are over, and it's well worth your time because it makes the science accessible. They really pride themselves on that, and they have great people behind the scenes collaborating with the artists and the scientists to make it into information bits that you guys can check out. So I highly recommend you go do that. It's way better than watching a movie. So the second thing in my Understanding Nature mission is to visualize information. And I wanted to figure out how I could do that and tell the story of the small scale science. So the story of the phytoplankton. The complication here is that phytoplankton are invisible to the naked eye. So we have to rely on instrumentation to see them. There's probably hundreds of thousands of plankton floating around this water that looks clear to our eye. I got to try even more instruments than what you just saw in that video. This is a flow cam, which did a great job of capturing the pigments and the quantities of plankton in the water. The resolution was a little pixelated for me because I really was going after the level of illustration done in the 1800s, which is actually quite hard to do when you're on a pitching and rolling ship. I also tried the microscope, and I'm not going to show you a video that I took a time lapse of this scenario where I'm bouncing all over the place. So I'm Viking, but I get seasick. So I was on Bonine 24/7, and I was trying to sketch and was able to do some awesome sketches, but there was no way I was going to be able to do a product on the cruise that involved a lot of plankton, which is what I wanted to do. I love equipment. This is the Cytobot. And the actual instrument looks like nothing, but it would take 5 milliliters of water every 22 minutes and sample the plankton in there. And we're talking plankton that is 10 microns so the diameter of your hair is 100 microns. So things 1/10 the diameter of your hair were popping up on the screen. This is an example. If I zoomed in, I could tell you when and where we were. But this is an example of the oligotrophic water, so not much. The black dots were a little spot on the camera that we couldn't get rid of, so just ignore the repeating black dots. So not a lot of diversity, but I was in love. I love the black and white instrument, what they produce, because I wanted to focus in on the shapes. Again, I was kind of emulating the 18th century scientists and artists, and I was really interested in looking at this from an artistic standpoint as well. So these all have timestamps on them. They all have scale bars. So I sat there and drew stuff, and we would have this very exciting moment when the new sample would come in, all the scientists would rush in, and I would rush in, and we'd look what was in the sample. This continued entirely across the Pacific. Then I started working on samples with watercolor. Again, working on a pitching rolling ship. I didn't know how big or small I could work. I figured it out. I definitely wanted black and white, and then I started doing rough layouts. So here I am actually cutting out photographs that were from the Cytobot and doing pencil sketches that are called thumbnails down in the lower right-hand corner because they're about the size of my thumb to try to figure out the design of this. So pretty standard in the scientific illustration, science communication, graphic design communities, you make a rough draft or what's called a preliminary sketch. So this was my preliminary sketch. Wasn't sure exactly how many plankton I wanted on it, but I knew I had to get drawing because there were like only 10 days left in this cruise. So I picked my plankton based on some of the feedback from the scientists and also based on what my personal favorites were. And this is an example of one of my sheets of, I would bunch them up on a watercolor paper and then I'd scan them in and clean them up and then go ahead and place them in InDesign. So Adobe InDesign is a graphic designer's application that I use to lay it out. So you can look at the close-up of the poster up here after. But I wanted to highlight that there are some really cool critters. And these are all phytoplankton actually, and coccolithophores, radiolarians. And true to what we're told, the art remains on the ship, or the art does find a home on the ship. So this is where my plankton poster, if you go do a cruise, if you go do a tour, you will see the plankton poster in the dining area. The third thing about my understanding nature is I wanted to try to capture the big picture. So I focused in on the big scale science, the big satellite imagery. Now, my eyes are not the same. First of all, I'm not out in space, but I was on a ship. But I wanted to compare and contrast what the scientists were capturing with satellite imagery and on the ship with radiometers, which is a way that they can look beyond the visible scale spectra and look at UV and infrared light. So this is an education information sheet that you can actually get off of satellites that are in the sky. The little gray boxes are the spectra that the satellites can actually grab from the visible light spectrum. And on the ship, we had 2 devices that were designed to measure the light and beyond the light spectrum. This was affectionately known as Batman. And it's a radiometer that went in the water. I'm doing on time here. Okay, I'm going to skip the video, but this is a video of it being drugged through the water. You can find this video online. It's sunk down to depth and it's taking live readings, and then we pull it back up on the ship. it was a very nice day when we took this footage. And then the other radiometer that they had is called the Hyper Sas Radiometer. It sits on the bow of the ship with the one angled up, and down at the sea with the ones angled down, and is measuring spectra. This is Ryan, one of the scientists, cleaning it, and I'm showing you this for scale because we did have rough days. If you look on the bow next to the tall pole, that's the Hyper Sas Radiometer. So the waves were not small. So again, the radiometers were taking microscale measurements of the ocean and the sky. The satellite imagery is taking pictures of the ocean and reading visible spectrum. The idea are that the pigments in phytoplankton can be picked up by these satellite images, and they can tell you something about productivity. So our eyes, as you know, can't see in the infrared or in UV without special aids. And I decided to emulate one of the scientists I found out, which he developed something called the Cyano Meter in 1760. I couldn't believe it when I found out about it. He used a pigment, interestingly enough, called Prussian blue, and he made 52 values of blue to measure the particle amount in the sky. It ended up not quite working out, but what was fascinating to me is that it was one of the first things I found out where they were trying to quantitatively assign something so that Joe Bob and Dr. Smith and Phil could all use the same cyanometer and say the sky was value number 42 today. So I made my own. It's hanging up out here. And I, every day, faithfully, during the almost 25 days of the trip, would go out on the deck, take a picture of the cyanometer. If you look at that, you can see that it actually kind of falls in line. You could, I don't know what it looks like up there, but on my screen here, it's about a 22, 23. So I did that every day and came away with 25 samples. Very qualitative. The lighting wasn't the same. I went out at different times. The weather was different. But the fact that I had 25 observations was really cool because it added something every day to my experience of understanding where we were on the planet, understanding what the conditions were, both sea state and sky, and also just making observations about weather. The second project I did that was tied to that is I wanted to make my own palette. I didn't want to use Saussier's palette. I wanted to make my own palette of blues and do a more creative project where I just qualitatively looked at the sky, looked at the sea, and wrote out a pretty sky and sea thing. So I called this Sky and Sea. The originals will be on display at the Bishop. I recorded the time, the date, and sometimes the conditions were a little bit windy. Let's see if I can play this. Oh, it's upside down. We didn't go upside down during the cruise. And I'm going to skip this one because we're out of time, but you can see this one online. This is on a really windy, horrible day where I did not lose a cyanometer, but I almost did when I was doing the filming. And this is what the final looked like. So again, set up like the cyanometer, it's 25 days. And these are very linked to the cyanometer in that I did them at the same time. So the time stamp on them is accurate-ish. But I love doing that because I made recordings like 5 albatross flew by while I was doing this. And probably the coolest thing where science turned back and used my art for a project is the NASA scientists are using my poster as part of their PACE program. PACE is a really cool thing they're doing where they're increasing, hopefully, the number of satellites in the sky and their sensitivity so that they can record to the same sensitivity that those radiometers can on ship. And that's really neat for me to see how they've used my artwork to help them explain their processes. I highly recommend that you look at PACE by looking up NASA and PACE, because NASA needs to look toward our planet, not just at Mars. So what I want to close with is if you guys want to explore anything, if we've improved, Michelle and I have improved the accessibility of this science through our art, you can look up the hashtag #ArtistAtSea and see our posts between Instagram, Twitter, and just on my blog. And then the Schmidt Ocean Institute, again, I mentioned this with the videos, but this is also true for the blog post that Michelle and I did. Excuse me. For the blog post that Michelle and I did, they are sitting there waiting to be absorbed. So they're out there on the Schmidt Ocean Institute website. So I encourage all of you to keep questioning, learning, and sharing like Michelle and I do, because we're science communicators, and we think everybody in this room can be science communicators by incorporating science and art into their life every day. And I just want to mention that she and I are continuing the artist adventure by going to Antarctica in 55 days. We got accepted as artists in residence at McMurdo Station for 7 weeks, diving under the ice to explore the science happening under the ice. And then one shout out to our other Artist at Sea friend, Lucy. Her stuff will be at the Bishop as well. And just in case you missed those dates from Carly, it's August 29th to September 29th. And we hope with that, you will come up and ask us questions and look at our stuff that we brought to show and tell. Thank you guys. Mahalo nui loa.